Hello students. Today we will discuss with another topic, cell cycle. Have you ever wondered how a tiny fertilized egg grows into a complete human body with the trillions of cells? The secret belongs to this cell cycle. Just like how we are following a daily routine, that is, we are waking up, then eating, waking up, eating is there, then working, resting, sleeping, like that we have a specific routine. Like that, even cell also having certain specific routine. They are following some routines, like they are having some growth will be there. The growth followed by there is a preparation. There is a preparation stages there for the duplication. Okay, so these different routine, that is the cell cycle. Okay, so that's what we are going to discuss in this topic. What is cell cycle? It is a ordered sequence of events. I have told you already, there is a routine following by the cell, that is the in a ordered way. Ordered sequence of events in the cell's life, involving the growth is there, then the DNA duplications, and finally the division of the cell into daughter cells. That whole process is called a cell cycle, right? So next, why cell need to be divided? There are certain purpose for the cell cycle. Very first important one is, we know that all the living organism, the fundamental feature is what? One of the fundamental features of living organism, that is the growth, ability to grow. And for the growth of the cell or the growth of the organism, cell cycle is very, very important. Another thing, if there is a wound formed in the body, this will, that wound will heal quickly. What is the reason? Because of the cell cycle. And for maintaining the genetic constant, genetic information and all, cell cycle plays an important role. So there are a number of significance for the cell cycle. So cell cycle is very, very important in all living organisms body. So in this topic, we will explore the different phases of cell cycle. So what are the various phases of cell cycle? The cell cycle, the whole cell cycle divided into two phases. One is the interphase and the second is the M phase. M phase is the actual division taking place. Okay, this is the phase in which the actual division taking place. The division means the division of the component, the whole cell. That means in the cell you can see there is a nucleus, and the nucleus will be there, and the cytoplasm, the other component and all. So the nuclear division, then the cytoplasmic division taking place in the M phase, that we will discuss in the next video about the M phase. This video we will more concentrate in the first phase, that is the interphase, right? So interphase, what is interphase? Interphase is the longest phase of cell cycle, and this is the preparatory phase. Okay, so interphase is actually the preparatory phase, preparatory phase, and this phase is also called a biosynthetic phase. Biosynthetic phase. Okay, so why it is called a preparatory or biosynthetic phase? Because in this phase only most of the metabolic reactions taking place and synthesis of all the components taking place in this phase. Okay, the whole phase has been divided into three substages, that is G1 phase, S phase, and G2 phase. So remember all the stages, G1, S, and G2 phases included under the interphase. And that is the longest phase. Okay, so in the cell cycle, the longest one is the interphase. Why it is called the longest one? I will give you with one example. In human body, okay, human cells, the parent cell divides into the daughter cell. So one cell divides into two daughter cells. Okay, and the generation of the parent cell into the parent cell into the daughter cell, that is called the generation time. Generation time. So the generation time in different cell it varies. For example, in human cells, the generation time is 24 hours. 24 hours taking place for the conversion of the one parent cell into two daughter cells. When you're comparing the whole phases like interphase and the M phase, 23 hours the cell will be in the interphase. Okay, and the remaining only 1 hour is for the M phase. That means majority time they will be spent for the interface only in the preparation time. As your exam is duration is very less only, final exams, right? But you have to study for a long time. Your preparations time is too much, and finally the exam will be in a very like a maximum 3 hours or 4 hours only. Everything will get over like that only, even in the cell also that is happening. Okay, so this interphase is the longest phase, consists of three substages, G1, S and G phase. Then in that stage the cell is metabolically very active, and there is a continuous preparation taking place, and all the other normal functions and all will be maintained by the cell. So let's discuss one by one the different subphases under the interface, right? So the G1 phase is first phase, and this is also called a gap 1 phase or first growth phase. Gap 1 phase or first growth phase. Okay, so first growth means the cells that intense growth will be there in this phase. The growth cells size will increase, and also the volume of the cell increases. And how the volume and all increases, because the duplication of the cell organelles taking place here. The different cell organelles like mitochondria will be there, that increase the duplicates. Then Golgi complex duplicates. Then the endoplasmic reticulum duplicates, like whatever the components, the organelles are there in the cytoplasm, the duplication taking place in the G1 phase. Along with that, the RNA, proteins, amino acids, ATP also will be synthesized here, right? And there is a critical checkpoint there. Does the cell have a sufficient resources there for the division, or the condition is favorable, that they will check in the G1 phase, right? Moving on to the next phase, that is the S phase. Okay, S phase is there, that is the DNA replication phase. That is the duplication of the DNA taking place. Whatever the parent nucleus, parent DNA is there, the content doubles in this. Okay, so the daughter DNA, there were from one DNA, two DNA strands produced. Okay, that means whatever the DNA, that content doubled during this phase. And also along with the DNA, there is a basic proteins there. That proteins are called histone proteins. So histone proteins also synthesized for the DNA package, right? So here important point you need to remember, the duplication of the DNA content taking place, and also the histone protein synthesis taking place. Now coming to the animal cell, there is a specific structure located near to the nucleus. It is called a centrosome. And the centrosome replicates during the S phase. One important point here, okay? Centrosome replicates during the animal cell. Related to this, there is a question. When you compare the G1 phase and the S phase, what are the number of centrosome? Number of centrosome present. Okay, number of centrosome present, and also the number of centrioles there. Centrioles present. Let's compare. G1 phase, that time in the cell there is only one centrosome is there. And so this is only the one centrosome. And each centrosome consists of centrioles. How many centrioles are there in each centrosome? There are centrioles there. Okay, so don't forget this. Every time there will be confusion with the centrosome, centriole. So G1 phase centrosome 1 will be there, and during the S phase the centrosome duplicates into 2. Okay, so after the S phase in a cell consists of 2 centrosome. So what will be the number of centrioles after S phase? It will be 4 centrioles, right? So moving on to the next phase, that is the G2 phase. G2 phase is also called a gap. Gap 1 phase is there, S phase is there, and then the gap 2 phase is there. This is the final preparation time. Okay. So, like your mock test and all, again, whatever you have learned in the previous month and all, everything, there will be final preparation. Like that, only here also, there is a final preparation. Again, there is a synthesis of cell organelles and all. Then the proteins will be synthesized. Little bit that what is that the spindle formation and all will be completed here. Okay, so there is a completion of the spindle proteins, microtubules, and also the centrosome duplication. Everything will be completed here, and also there is a checkpoint. Okay, so whatever the DNA replicated in the previous phase, that is the S phase, whether it is correctly replicated, is there any errors happened, that if the error is there, that repairment, everything will be happening in this G2 phase, right? So remember all the three phases under this interphase. What are the three phases? Yes, G1 phase, S phase, and G2 phase. Okay, G1 phase, S, and G2 phase. We are coming back to the diagram again. Just have a look at the diagram. So here you can see there is a growth and a normal metabolic roles there in the G phase, followed by S phase. Here the crucial step is the DNA replication taking place, and the final preparation, again the growth and the preparation of final preparation for the M phase taking place. Okay, sometime some cells and all, they quit the cycle just at the half of the G1 phase. They quit the cycle and they will go out, and they will be in a resting stage, and that is called a G0 phase. Okay, so we need to discuss a little point about the G0 phase also, some points only. So what is G0 phase? Okay, so G0 phase is that the certain cells, they quit the cell cycle, they will become like inactive form. Okay, inactive form means they are metabolically active only, but they are inactive for the cell division. There is no cell division and all, but they are metabolically active. Okay, such kind of phase is called, the cells will go to the phase is called a G0 phase, right? So in that time, what happens, the some that the time spent ranges from some cells, it will be only hours only. Okay, the G0 phase, some cells and all, permanently they become in the G0 phase, that depending upon the type of cells or the signals they are receiving, depending upon that only the duration of the G0 phase will be determined. Okay, so when you think about our neurons, we know that the neurons, once the division happens after the cell, after the birth, then there is no more cell division from the parent neurons. Okay, so the neurons remain permanently in the G0 phase, and they will perform a specific function. We know the function of the neuron for the transmission of impulses. So they will do only that function. There is no more production of the new neurons there. Okay, so this G0 states, zero phases will be in the different conditions, a different stage you can see. Some cells, the G0 will be like a reversible resting state. That means when they are getting enough signals and all, they will come back to the cell cycle. They will go back to the cell cycle process. Okay, some cells are and all in the irreversible arrest. That means completely they stopped the cell cycle. Okay, and that is due to certain reasons, and maybe there is a damage in the DNA or stress or cellular aging and all that causes the senescent G0. Okay, so one is the quiescent G0. Quiescent G0 phase is the reversible resting state, but senescent G0 is a irreversible arrest. Okay, and the third one is a differentiated G0, like a neurons or that muscles and all. We can see red blood cells and all. We can see that they exit from the cell cycle and they perform a specific function. Okay, they will not go back to the cell cycle, but they have a specific, they are assigned for some specific functions and all. That's what in the differentiated G0. I hope you understood this concept regarding this cell cycle, interphase, different stages and all, right? So remember certain points in the interphase state. The DNA exists in the loose thread like a chromatin, only in the interphase stage, not in the form of chromosomes. It is in the form of a chromatin only. But post replication, replication happened in the S phase. After the S phase, when you check, the chromatin threads are there, but there you can see sister chromatids. Okay, not the number of chromosome will be same only, but they have a two sister chromatids pair that will be there, and they joined in the with the help of a specific protein. And what is its name? It is called a centromere. Called a centromere. Okay, remember the term centromere. Okay, so let's discuss some questions related to this. I need to check your understanding of the concepts. So there are some 5 questions. We'll discuss now. First question: in eukaryotes, the cell cycle is divided into which? What is the correct option? It is divided into G1, S, G2, and the M phase. Okay, this is the whole process. So we can remember with the mnemonics. What is the mnemonic for to remember this one? Go study, go study, gain marks. Go study, gain marks. G for G1, S is for the S phase or synthetic phase, G for the gap 2 phase, and M is the M phase. Okay, so the correct answer is the option A. Second question: which phase of the cell cycle is known as DNA synthesis phase? Very easy, right? It is the S phase. Third question: a cell with the 46 chromosome enters in the S phase. At the end of the S phase, it will have dash number of chromosomes. We know that during the S phase, there is a synthesis of DNA taking place, but not the number of chromosome will not increase, only the content doubles, right? So the number of chromosomes will be same only even after the S phase also. So the answer is option B, the same number of chromosomes there. Then the fourth question: which checkpoint ensures the DNA replication is complete before the mitosis begins? That is the completion, whether the replication is completed, is there any error happens, everything will be checked in the G2 phase, right? The last question, but not the least: the resting phase or the non-dividing state of the cell cycle is known as which one? That is the G0 phase. Okay, I hope this topic is clear for all of you. Thank you. Have a great day.